And now it's my pleasure to introduce to you Grace Russell, who I've gotten to know over the years. She's very active in the molecular hydrogen research and publishing articles. We've collaborated on a number of articles now. She always has excellent questions and thoughts, and she's works really hard. She's actually one of the first ones to take the MHI Level 1 certification course and even provided some helpful feedback and things. So I appreciate her efforts and what she's trying to do and everything. So thank you, Grace. Thank you for supporting this MHI Summit. Now I'll read her bio. Grace Russell is a driven and accomplished researcher specializing in biomedical sciences with a particular focus on molecular hydrogen and oxyhydrogen gases. She graduated from the University of West England in 2020, earning her Master's of Science degree with distinction. Currently, Grace is immersed in her doctoral research at UWE, exploring the effects of these gases, with plans to graduate in summer of 2024, which she has now graduated, being September. She's completed that. Congratulations, Grace. Grace's academic journey is marketed by excellence and dedication. Despite being early in her career, she has already made significant contributions to the field. Grace's scholarly prowess is evident from her numerous publications in esteemed peer-reviewed journals. Beyond her research, Grace is passionate about advocating for intersectional representation and ensuring the voices from diverse backgrounds are heard and valued in the scientific community. And now please welcome Grace.
Hello, my name's Grace Russell, and I've recently completed a PhD in hydrogen biochemistry at the University of the West of England, Bristol. Welcome to this section of my presentation, which focuses on the evolutionary role of molecular hydrogen, investigating whether orthologs of hydrogenase enzymes exist within the human proteome. Understanding whether relics of hydrogenases persist in our genome could reveal new insights into our evolutionary past and the potential roles hydrogen and its metabolism may have in human biology. Around 4 billion years ago, Earth's primordial atmosphere was rich in hydrogen and hydrogen-containing gases such as methane, thereby setting the stage for the formation of basic organic molecules. From an evolutionary standpoint, it is likely that the dissociation of molecular hydrogen, abundant in the atmosphere at the time, was fundamental to primitive cellular processes, including energy production through the generation of chemiosmotic gradients, a basal function used in all domains of life today. Evolution of the eukaryotic lineage is a highly contentious topic that spirits much debate amongst academics. Contemporary thinking reveals 5 main theories of cellular evolution. These are the reverse flow model, involving an organotrophic archaeon and an acetate formate producing alpha bacteria; Cersei's model, involving a sulfate reducing archaeon and a sulfate producing bacteria; the E3 hypothesis, that builds on the reverse flow model, suggests that an archaeon alpha bacteria duo were joined by a delta bacterium that utilized the hydrogen waste products of these early life forms. Although the hydrogen hypothesis, involving a methanogenic archaeon and a methane-reducing alpha bacterium, suggests hydrogen has a major role in eukaryotic evolution, it is the syntrophy theory that suggests that a delta bacteria utilize both an intracellular archaeon and an alpha bacteria that shape this inquiry. Hydrogenase enzymes, found in all single-celled and some multicellular organisms, are responsible for catalyzing the reversible dissociation of molecular hydrogen, generating protons and electrons. However, the group 4 membrane-bound hydrogenases share sequence similarities with specific subunits of mitochondrial complex 1 and are of interest here. Group 4 nickel-iron hydrogenases comprise of a small cytosol-facing unit responsible for shuttling hydrogen molecules to the larger membrane-bound unit that houses the deprotonation or active site. This is similar to the configuration of complex 1, the first protein of the energy-producing electron transport chain within the mitochondria. For this research, I focused on the potential evolutionary relationship between the redox active subunits of complex 1 and 3 microorganisms possibly involved in the syntrophic hypothesis of eukaryotic evolution. These microorganisms are Delta Proteobacterium desulfovibrio carbonolicus, the host, an Asgard archaean, Candidatus hemdala archaeota, that may have formed the nucleus, and an alpha proteobacterium, Rhodobacter spheroides, possibly the ancestor of the mitochondria. To gain a better understanding of potential similarities between the nickel-iron hydrogenases of such microorganisms, several bioinformatic methods were applied. Firstly, a BLAST-P analysis of sequences helped to identify whether there are orthologs in the human proteome. Next, to visualize homology, a matrix alignment analysis was performed. Thirdly, a sequence alignment was used to assess the level of sequence congruity. And finally, to identify whether the regions of similarity were likely to be functional, ScanProtiste, a library of biologically meaningful signatures, was used. The BLAST-P analysis identifies sequence similarities between the hydrogenases of all 3 organisms and 2 subunits of complex 1, known as NDUFS2 and NDUFS7. Interestingly, these 2 subunits form part of the ubiquinone binding pocket and are known to have redox activity, being the primary units responsible for ubiquinone reduction. When considering complex 1 is the primary source of reactive oxygen species, and that the primary sites of ROS production are the flavin mononucleotide unit here, and the ubiquinone binding site here, the fact that these units show similarities with hydrogenases is intriguing, so I started looking deeper into these affinities. As the large hydrogenase subunit that corresponds with the human NDUFS2 protein houses the hydrogen deprotonation site, this was the first region to be explored. The cluster alignment showed some interesting things. Although there are few similarities between species, the redox-active residues arginine, glycine, and glutamic acid are conserved. But perhaps even more interesting is that the tyrosine amino acid 151 in humans, thought to be responsible for electron transference, is conserved in the hemdala archaeota, suggesting this site could be crucial not only for ubiquinone reduction, but perhaps, considering the potential for electron leakage and ROS production, this region could also be a target for hydrogen. Although the region around the active site shows little homology between species, the matrix analysis identifies matching sequences between proteins of hemdala archaeon and NDUFS2, noted here by the diagonal lines and pink arrow. And further analysis shows that histidine 87 and asparagine 179 in humans, which may have proton translocation potential, are well conserved. This may be an important factor, as it is possible that these residues could work collaboratively, creating a negative charge that would lower the redox potential of the N2 iron-sulfur cluster, perhaps permitting reverse electron transfer. The matrix analysis of desulfovibrio carbonolicus shows a much less obvious association between NDUFS2 and the large hydrogenase subunit. The cluster alignment supports this, with only histidine 87 of the potential redox triad being apparent in both proteins. Rhodobacter spheroides has 2 nickel-iron hydrogenases, and both were investigated here. The matrix analysis shows very little homology between species, a factor that is again reflected in the sequence alignment, where none of the key amino acids, tyrosine, histidine, or asparagine, are conserved. So far, we can deduce from these findings that the large hydrogenase subunits of either bacteria have little homology with the NDU FS2 subunit of complex 1. But what appear to be key redox residues in the archaeal hydrogenase are indeed well conserved. Things become a little more interesting when I began to look into the similarities between the smaller hydrogenase unit and its potential human counterpart, NDU FS7. With Haemdalla archaeosa, again we see a strong correlation between species, indicated by the dark line and arrow. The clustral alignment reveals some interesting matching too. For example, the RASP-RQS motif, highlighted in blue, is a target for protein kinase C phosphorylation, and we can see here that this section is reasonably well conserved. A similar level of conservation is seen further along the sequence at the IPG-CPP motif, which is a site for N-myristylation, a lipid modification to the glycine residue. This post-translational modification has a crucial role in regulating protein function, influencing the protein's stability, activity, and its ability to interact with other cellular components. Should hydrogen have effects in this region, it could also affect ubiquinone binding, thereby reducing the likelihood of electron leakage and ROS formation. With desulfovibrio carbonolicus, we see another, if somewhat weaker, correlation between proteins. Again, we only see part of the phosphorylation site as sharing sequence identity, but curiously, the N-myristylation site is fully conserved, indicating this region may be of importance to protein and perhaps electron shuttling activity. In Rhodobacter spheroides, there is no sequence alignment with the protein phosphorylation motif, RASP-RQS. However, the IPGCPP site is once again well conserved, with the key glycine residue being present in all organisms studied here. Such residues may be of pertinence as the sites of hydrogen activity, as hydrogen may stabilize the protein structure, protecting such sites from auto-oxidation. This is purely speculative, however, and far more research will be needed before we can be sure that the influence hydrogen may have on the activity of complex 1. In conclusion, this research has identified a possible evolutionary relationship between redox-active subunits of complex 1 and both archaeal and bacterial nickel-iron hydrogenases. Among the microorganisms investigated here, the strongest candidate for an evolutionary link is with Candidatus Haemdalla archaeon. This discovery supports the syntrophic theory of eukaryotic evolution, which suggests that the cooperative interactions between different microbial species were crucial for the emergence of eukaryotic cells. Understanding the evolutionary relationship of these proteins may highlight how more complex organisms harness molecular hydrogen to improve metabolic processes. Here are the references used to make this presentation.